Good morning, everyone. How are you, students? Okay, good morning. Good morning. Okay, so students, earlier taught you rain problem and riverboat problem. Okay, you recognized me? Okay, students. Thank you, thank you. Okay, so in last class we understood relative motion, everything, and before my class, Paul sir taught you about motion and Newton's laws and forces also. He explained about forces, how many forces are there in the nature, everything. Okay. So today we will understand about momentum. So today our topic is linear momentum. And this topic is very important. This topic is very important because momentum is the basis of motion. How? If a body has motion means velocity. So you can say, sir, velocity is important, not momentum. But I am saying momentum is more important than velocity. How? Suppose just example. We have two bodies and one body mass is 1 kilogram. Okay. Just leave the body, just a normal example. Suppose one is elephant. Elephant is moving. Okay. And elephant is moving with velocity 5 meter per second. Okay. From opposite way, you are moving and your mass is I think 55 or 60 kilogram. Okay. So from one side elephant is moving, from another side you are moving and your mass is 60 kilogram. Speed is equal. Elephant speed 5 meter per second, your speed also 5 meter per second. Okay. If both will collide, then what will happen? You will say, sir, nothing happened for elephant. Whatever it will happen, that is, it will affect only us. Okay. So, what is the difference here? Elephant velocity and your velocity equal, but no effect on elephant. That collision affect only you. Why? Because your mass is less. Right or not? Some students are writing RIP. Great. Always think positive. There is a chance maybe you will survive. Okay. So important point is what? Not only velocity play role. Velocity with mass. How much mass has how much velocity, both play important role. Different example. Some students are asking offline, when offline classes will start. Offline classes will start soon. Okay, yesterday NEET paper conducted, and this time from our center, from this Chaitanya Gosala, students are getting very good marks. It is historical result we are achieving this time. We are breaking our past records and this time it is really amazing result we are getting. And I want to give an example also, because till now I do not know all the students marks. Only some students called me and they told their marks. So this time, a normal example I want to give. In 11th class, one student was there, one girl was there. She was getting 90, 100, 110, 120 maximum. Like that marks she was getting in physics. Okay. Then in 12th class, she started to getting good marks. Okay. 120, 130 also she got. 140 also she got in some exam, only in some exam, because you know here every week one exam conducted. Okay. So she was getting good marks like 130, 140, in some test less also, 100 also, 110 also, okay, like that. But finally, will you believe how much marks she got in yesterday NEET exam? Can you guess? Send your response. Can you believe? This is, you know, I am giving this example only because it is really inspiring. Okay, some students are sending 180, some are sending 170. No, she is getting 162 marks, not 180, not 170. She is getting 162 marks in physics. 180 this time only a few students are getting, because in section A, two sections are there in the exam. In section A, total 35 questions are there. Out of those 35, 2-3 questions are really difficult. Time taking problems, means if you solve them, so at least you have to give 2 minutes, then only you will get answer. Like that questions were there. So due to she actually left that question that I will solve finally, if I have time then I will solve, but she didn't have sufficient time, that's why she left those questions. So due to that she got 162 marks. So this is also, you know, very inspiring. In 11th class 100, 110, like that she was getting. In 12th class also this 130, 140 like that, and 100 also. But finally she did blaster 162 marks, right? So students, every time I say that always do your own analysis, where I am standing now, where I was. If you are doing well in present then past, then 100% in finally also, in final exam also, you will get very good marks. Okay. Am I audible? Some students are saying that, sir, be louder, like that. Audio, video clear? Am I audible? Okay, okay. So today, return back to our topic. Our topic is linear momentum. So we understood one point, that only velocity will not play the role. Velocity with mass both play the role. I given one example of elephant and you. Now another example. One side you fired a bullet and your target is elephant, suppose. Bullet mass is maximum 100 gram, maximum, or maybe less. So mass is 100 gram for bullet, and mass of elephant very higher, maybe 1000 kilogram, 1500 kilogram, 2000 kilogram, like that. Okay. To one side very heavy object, 1000 kilogram, and another side bullet, mass very less. If you compare this 100 gram with respect to 1000 kilogram, then it is very less. Okay. But we fire a bullet, means bullet velocity is very high. Generally, if we take bullet velocity, then it is around 100 meter per second, 150, 200 meter per second. It depends on gun. Which gun you have, right? AK-47, you listen the name, right? In movies. Okay. So that bullet velocity is around, suppose, 200 meter per second. So what will happen now? Maybe there is a chance that bullet can cross completely through the elephant. I'm not like that person. I'm not a hunter. Okay. but I just given the example. So this time what play the important role? Velocity, right or not? Agree or not? So earlier I given example in which mass, and this time velocity, right? So in motion both play important role, mass and velocity. Is it clear? Okay. So students, we understood that a quantity we should define. You know how physics started? That I am explaining. Like that you have to think. When you start to think like that, then your physics marks will be 160, 170, 180 out of 180. Okay now? So here we understood, some students are they know the formula. Okay, formula is P equal to mv. Formula you know, but understand that how those scientists who worked first on momentum, how they think about momentum, that I am explaining. Okay, so we understood that a quantity we should define. There is a requirement to define the quantity in which it has both mass and velocity. That quantity is linear momentum. Clear? So this linear momentum formula is mv. Okay, product of mass with velocity. So definition is product of mass with velocity is linear momentum. So formula also we understood, P equal to mv, m is mass and here v is speed. Okay, now understand this point. You studied that there are two type of quantities, one is scalar, another is vector. So this quantity is which one, scalar or vector? So this quantity is vector quantity. It is vector quantity. Now you know, in vector quantity two things are required, magnitude and direction. Magnitude and direction. So its magnitude is mv. So when you are writing magnitude at that time, what is v? V is speed. Is it clear? So what is p here? This is magnitude of momentum. This is magnitude of momentum. Is it clear? Important point it is. Okay, I am not writing here velocity, I am writing here speed. Why? Because if you are calculating magnitude, then in case of magnitude you have to take here speed. Understood? Okay, now what is direction? So we understood that it is vector quantity, so it has both magnitude and direction. So its magnitude is mv and its direction now. So its direction is along the velocity. So what is the direction of momentum? Along the velocity. So second point, its first its magnitude is mv and its direction is along the velocity. So we understood this point also. So both point we understood, magnitude and direction. Now here what you understood, that if in physics, very important point it is, if in physics anywhere you see vector quantity, so in that vector quantity, like that you have to split into two parts, that how much its magnitude, how much its, and what is its direction. Is it clear? So magnitude we understood and direction is along velocity. Now in case of vector quantity, sometimes they ask that write its vector form. So sometime we have to write its vector form. So how to write vector form? Understand that one also. Vector form of momentum. So vector form we write like this: P equal to mv. This is vector form. Now in this vector form, if you can ask, sir, what is v? This is now velocity. This is not speed. Is it clear? So what is m here? M, okay, mass, but this v vector is velocity. It is not speed. This is the difference. And this is linear momentum vector. This is linear momentum vector. Understood? Understood, friends? Is it clear now? So I split in both part. One is direction and one is magnitude. Direction is along velocity, magnitude is mv. Okay. And here we understood magnitude formula this much, vector form formula this one. Okay. Now I am asking from you, what is the unit of momentum? To send your answer, I will see who will send first. Send answer unit of SI unit of momentum. SI unit of momentum. Send your answer. Okay, Sushant, very good. First this boy sent the answer, correct answer. Then the Viber, this Viber, earlier also I asked what is your correct, exact name. Okay, Dheeraj and this Garuda. Who is Garuda? I seen one movie. Which movie that is? KGF. KGF. In KGF, I heard this name Garuda. Okay. Now all these friends are sending answer and correct they are sending. Very good, very good, very good, very good. All are sending correct answer. Very good. SI unit of momentum. SI unit of momentum is, so how to find SI unit? You know, this is simple, okay, but sometimes they ask SI unit for some complex quantities. So where we do mistake? Why we do mistake? Because we do not know exactly that how to calculate. So how to calculate? Very simple. You have to find out unit of, each quantity that is in the formula. Now which formula? Some students can ask, sir, this formula or this formula? Very important point, punch point. What is it? That whether the quantity scalar or vector. Suppose momentum we are talking. Momentum is vector, but we have its scalar form also. This is scalar form, this is vector form. Okay. So in such quantities, actually unit is equal for vector form or scalar form. Very important point. Unit does not affect by scalar form or vector form. If we talk about velocity, vector, speed, scalar, velocity unit meter per second, speed unit meter per second. So very important that dimension and unit both are unaffected from the type of quantity, that it is vector or scalar. So this point is notable. Okay. So SI unit of momentum, we can use any formula. Mass SI unit you all know kilogram, and velocity SI unit you all know meter per second. So SI unit of momentum is kilogram meter per second. Kilogram meter per second. Clear? Okay. So we understood about momentum. Now here actually we taken vector form. Vector form means here direction is involved. Direction is involved means suppose a body is moving in x direction, so x direction momentum, y direction, then y direction momentum, z direction, then z direction momentum, right or not? So momentum can be in x direction, momentum can be in y, z or all direction. It depends on the velocity of the body. Is it clear? Okay. So here if we talk about the direction of momentum, so momentum for general motion. General motion means we have to assume that body is moving in all direction x, y, z. So P net equal to how much? M P net equal to how much? So it is in x direction, it is in y direction, it is in z direction. So x direction, then, and here we can write I cap also. X direction, Px, it is in x direction, and you know for x direction which vector we use? I cap. Okay, and in y direction Py, which vector we use? J cap, and in z direction Pz, which vector we use? K cap. Is it clear? So this is net momentum we have to write for a general body which is moving in all direction x, y, z. Now you can here write the formula. Px formula is mvx, right? Py formula is mvy, and pz formula is mvz. Okay, so here px means x direction velocity we have to take, so mass multiplied with x direction velocity. In mass, complete mass we have to take. Don't think that in mass sir x, y, z. No, in mass complete mass. mvx, m y direction velocity, m z direction velocity. Clear? Okay, so we understood about momentum. Theoretical part we understood. Now we will understand very important point, conservation of momentum. Conservation of momentum. It is very, very important, and today in this class, listen this class very carefully, because today in this class we will solve NEET previous year questions. So now you can realize that how important this class. After listening this 15 to 20 minutes, you are able to solve NEET questions, NEET previous year questions, right? So don't think that array it is online class. Whatever we are teaching, that is very important. It will play very important role in this coming 11th and 12th both years. Clear? Okay. So next we will solve question, but before that we have to understand this point, momentum conservation. Momentum conservation. Understand it. You all studied Newton's laws. Can you tell what is Newton's second law? Send your response, I am waiting. What is Newton's second law? One student, this Sheikh Fahad, he sent that the dimension formula of momentum is m1 l1 t minus 1. Very good, very good. Yes, it is correct. Very good. Garuda is sending, I think I am loving to do physics. Very good. Your real name also Garuda? Okay. So all students are sending that Newton's second law is F equal to ma. all are sending that Newton's second law is F equal to ma. One student, this Himanya, she sent that rate of change of momentum is directly proportional to external unbalanced force. Very good. This is, out of all the answers, this is perfect answer. Okay, very good. Okay, Garuda sent that his real name is Rian. Okay, I will remember your name. Okay, so momentum conservation we are understanding. So I asked you that what is Newton's second law. Some students are sending, most of the, not some, most of the students are sending that it is F equal to ma. But in reality, it is not only F equal to ma. It is what? Definition, you know. Definition is rate of change of momentum is directly proportional to force. Which force? External force. Means only external force can change the momentum. You agree or not? In definition only, very important point. Again listen. Second law is rate of change of momentum with respect to time is directly proportional to applied external force. So if we apply external force, then only momentum will change. If we will not apply external force, then momentum will not change. Agree or not? You all agree or not? Okay. So if external force 0, then no change in momentum. Yes, okay. If no change in momentum means whatever now value of momentum, that will be remain equal for next 5 seconds, next to next 5 seconds. Agree or not? Means whatever the quantity value now, it will be same after some time, it will be same after some time. Right or not? This is known as momentum conservation. If the quantity value remain constant, it is not changing, means it is conserved. You have 100 rupees, now after 1 year also 100 rupees, after 5 year also 100 rupees, so you can say that in 5 year span to my amount is fixed, constant, conserved, you can say. Right? So what is momentum conservation? That momentum value remain constant, it will not change if the external force is 0. By second law only we understood. Okay, so definition is if external force is 0, then total momentum of the system remain conserved. Clear? And from where it came? It came by second law. F external is delta P by delta T. This is external force. So if this F external is 0, then we can say if it is 0, then this is also 0. If it is 0, so this is also 0. Yes. So delta P is 0. What is delta P? Delta P means change. Always remember friends, delta means change. This sign delta na, it means change. Change means final minus initial. Okay, so delta P means P final minus P initial, it is 0. So we can say that P final equal to P initial. Understood? Okay. So final momentum equal to initial momentum. This is what? This is momentum conservation. It is known as and here we will write system. This is for system, not for a single body. It is for system, and this is known as momentum conservation. This is momentum conservation. So first point, when it will remain conserved? If external force is 0. So these two conditions are important. If external force 0, then it will remain conserved. Conserved means final equal to initial. So here you can break what? If general motion you take here, means motion is in xyz direction. So you can say that final momentum in x direction equal to initial momentum in x direction. Final momentum in y direction is equal to initial momentum in y direction. Final momentum in z direction is equal to initial momentum in z direction. You can break like this. Is it clear to all of you? Understood? So we understood momentum conservation. Now where to apply momentum conservation? In which questions it will remain conserved? So if they ask explosion question, a rock is there and suddenly it explode. So you applied any force on that? No, you are not there. Okay. Naturally it explode, means it explode due to internal force. Externally we are not applying any force on the rock. So in explosion questions, momentum remain conserved. First point, we will solve NEET questions now. So first point, where it will remain conserved, you have to remember it. Why? Because today only we will solve question. So in explosion, momentum remain conserved. Very important points these are. In explosion, momentum remain conserved. Second is bullet gun problem. You have gun, okay. Now you just trigger, है ना? What will happen? Bullet will come out. You applied any force on the bullet? No. Whatever happened, why bullet will come out? Due to internal action. So whatever happened, that happened inside the gun, right or not? So in bullet gun problem also momentum remain conserved. In bullet gun system, momentum remain conserved. Clear? Okay, we understood that in bullet gun system momentum remain conserved. In explosion momentum remain conserved. Okay, other are also there, but today we will discuss only on these two topics. We will solve question. Okay, to just see it how to solve questions. If this system will come, then what you have to do? Final momentum. Suppose if they ask explosion, so before the explosion whatever momentum equal to momentum which is after explosion. Bullet gun they ask, means before firing the bullet, whatever is the momentum, that momentum must be equal to momentum which is after firing. Understood? That is the meaning of conservation here. Initial equal to final. Initial means before action. Before explosion initial, after explosion final, must be equal. In bullet gun Before firing initial, after firing final, so both must be equal. Is it clear? Okay. So we understood it. Now we will solve question. So this question we are solving this question. This question came in NEET 2019. Okay. How to solve this question? What is given? An object flying in the air with velocity, this velocity is given. Okay, so what is given? Initial velocity is given. Okay, perfect. This is what initial velocity. Now, by using this initial velocity, what we can calculate? What we can calculate? We can calculate initial momentum. Agree or not? So, initial momentum in x direction, how much? mv. Right? In this is what vx, x direction velocity. This is what vy, y direction velocity, and this is what vz, z direction velocity. Agree or not? Okay. So this is z direction and this is y and this is x. Okay. Suddenly breaks into pieces whose mass are in the ratio 1:5. 1:5. Okay. The smaller mass fly off with velocity, smaller mass velocity also given. Okay. Students are, it is not clear, eh? Is it? It is not visible. Okay. So I am reading the question. I am writing the values, whatever given. I am writing. Just listen. Okay. So what is given? Initial velocity. Listen the question. An object flying in air with velocity. They given the velocity. How much velocity is given? I am writing it. V equal to 20 I cap plus 25 j cap minus 12 k cap. This velocity is given. This is what? Initial velocity. Okay. Suddenly breaks into two pieces. Now this object, whatever it was flying, it breaks into two pieces. Okay. Whose mass are in the ratio 1:5. So breaks in two pieces. Okay. Breaks in two pieces. Mass ratio is given how much? 1:5. 1:5. Okay. Smaller mass fly off with velocity. To smaller mass velocity is given. Smaller mass velocity is given means it is after collision. How much? 100 I cap plus 35 j cap plus 8 k cap. This is smaller mass velocity. Velocity of larger piece. We have to find out velocity of larger piece. So how we will calculate? Same concept we have to apply. Initial momentum equal to final. But in each question we have to see option. Now see the option. Very interesting point is what? That here in the options, I cap, see I cap, in I cap how much velocity is given? So here 4, here 20, here minus 100 and here minus 20. So you can see here in all option, I cap velocity is different. Agree or not? In all option, I cap velocity is different. Okay, now focus on this question. When some students are asking, this Jyoti is asking how was yesterday NEET exam. I already talked about NEET exam, na? I already talked about everything about this NEET exam. So now we have to focus on this question, how to solve this question. So they are asking the velocity of larger piece, and in option I am saying that how to solve. Don't just after reading the question, don't start to solve the question. No, see the option every time. This is the best strategy that we have to follow. So in all option, I cap, direction, velocity are different. So we have to calculate only I cap direction velocity. Agree or not? So in I cap means x direction. So in x direction, whatever initial momentum equal to final. Okay. Now this body breaks into two part, 1:5. If it breaks into two part, 1:5, so we can assume that this one part mass is m and another part mass is 5m. Right or not? Now take ratio. M ratio 5m is how much? 1:5. So we have to assume that its mass is m, then its mass will be 5m. So total mass how much? Total mass is 6m. Agree or not? Okay. So total mass 6m. Now initial momentum in x direction how much? Initial momentum in x direction how much? So we discussed already that how to write it. So in x direction it is mv, which v x, that also we understood. We discuss here, we discussed that initial momentum equal to final. Initial equal to final in x, y, z like this. And momentum value we have to write mv, that also I already explained you. Here momentum in x means mvx, y means mvy, z means mvz. Okay. So mvx. Now m is how much? Initial we are calculating here. Initial means before explosion. Before explosion total mass is 6m. So it is 6m into vx. So initial velocity is how much? 20. And it is I cap plus and plus 20. Okay. So this value we calculated. It is how much? It is 120 m. Now calculate final. In final, how many, I wrote there system. You have to find momentum of system. So finally means after explosion. So after explosion, how many mass we have? 2 mass. So we have to calculate momentum of each mass. Okay. So mv x, m1 v1x and m2 v2x. So m1 means smaller mass m. Its velocity, to smaller mass velocity in x direction how much? 100. So it is 100. Okay. Plus this second mass, this is larger. How much it is? 5m. So 5m into velocity that they are asking how much velocity in x. So we assume that it is vx. This is final momentum. So now we will put initial equal to final. So initial momentum in x direction equal to final momentum in x. So initial how much? 120 m. And final how much? 100 m plus 5 m vx. Transfer this value this side. 120 minus 100 how much? 20. So it is 20 m equal to 5 m into vx. M cancel out. 20 by 5 4. 20 by 5 4. So vx equal to 20 by 5. So our answer is 4. Now check in which option x direction velocity is 4. So here 20 minus 100 minus 20 to only in one option, that is first option. Agree or not? So in first option velocity in x direction is how much? 4. We are also getting 4. So this is our correct answer. Yes, you are sending correct. Most of the students are sending A option. Yes, correct. Okay, very good. They ask in all direction, but why I calculated here x direction? Only because in all option x direction velocity is different. Okay, so only we have to calculate x direction velocity. This option is matching, means this is our answer. Is it clear? Understood? Okay students, so today We understood about linear momentum. First we discussed theoretical part, examples we discussed, where to apply momentum conservation that we discussed. Then we solved questions, and in front of you I solved this question. It came in NEET exam. So you can see that it is not difficult. Physics is a simple subject. Okay, so okay, bye bye students. I hope that all you enjoyed my class. Now chemistry sir will take class.
Hello, good morning, sir. Good morning, students. So today we are going to discuss next new topic of organic chemistry, that is degree of unsaturation. Now, why degree of unsaturation is very important? So when we study isomerism, that is structural isomerism, where they will ask a molecular formula is given, draw all the possible isomers. So there we should know that how many possible structures are there. That compound is cyclic compound, compound is open chain compound, how many double bonds are possible, how many triple bonds are possible. So for that, what is required? Degree of unsaturation is required. So it is a very good topic, interesting topic, and useful for isomerism also. So what we write, heading is degree of unsaturation. In short form, what we write this as du. And there are some other names for degree of unsaturation also. Next name is, it is double bond equivalent. Another name for degree of unsaturation is double bond equivalent. So in short form, what we write, DBE. And one another name for this is hydrogen deficiency index. Hydrogen deficiency index. In short form, what we write, HDI. So all these names you should know. For example, if they ask calculate degree of unsaturation for this compound, it is very easy if you know this concept. But sometimes they will write what is double bond equivalent for this compound. Now I think we know degree of unsaturation. Now what is this double bond equivalent? All these names are same. Degree of unsaturation is also called as double bond equivalent, or it is also called as hydrogen deficiency index. Now what is definition of this degree of unsaturation? So listen students, when there are two types of compounds, one is saturated and one is unsaturated. So what is meaning of saturated compounds? Which contains only single bond, that are called as saturated. And compounds which contain at least one double bond or triple bond are called as unsaturated compound. So if we have to convert unsaturated compound to saturated, we have to add hydrogen. So that double bond converted to single bond, hydrogen is added. Triple bond converted to single bond, hydrogen is added. So how many moles of hydrogen is required to convert 1 mole of unsaturated compound to saturated compound? Or we can say 1 mole of cyclic compound to open chain compound. So this is called as degree of unsaturation. So why it is called as hydrogen deficiency index? Because it represents that how many moles of hydrogen are deficient for which type of compound, saturated compound or open chain compound. So if we write definition, it is defined as, so this definition is very important. Number of moles of hydrogen required to convert 1 mole of unsaturated compound to saturated compound. Or we can say, or 1 mole of cyclic compound to open chain compound. So this is called as degree of unsaturation. So what we have to do? We have to convert 1 mole of unsaturated compound to saturated. So by balancing we can know how many moles of hydrogen are required. That will be called as its degree of unsaturation. Or for 1 mole of cyclic compound to converts into its homologue open chain compound. Means same number of carbon atoms will remain same. So cyclic compound will be converted to open chain. So how many moles of hydrogen are required? That is called as degree of unsaturation. Now we will take examples for this. So let us take first example is very simple compound C2H4. What is name of C2H4? C2H4 is ethene, and we know it is carbon, double bond carbon, hydrogen, hydrogen, hydrogen. Here, now which type of compound it is, saturated or unsaturated? You have to tell student this compound is saturated compound or unsaturated compound. Now this is a unsaturated compound. Why cyclic compound? We'll take example of cyclic also. Wait for 1 or 2 minutes. First, this is a unsaturated compound. Now how many moles we have to take? 1 mole. Now if we add hydrogen to it, what is unsaturated compound formed will be CH3CH3. So what is formed here? Ethane is formed. Now if you check, this reaction is a balanced reaction. C2H4 plus H2 gives C2H6. So how many mole of hydrogen are required for 1 mole? 1 mole of hydrogen is required. So it means what is degree of unsaturation for this compound C2H4? It is equal to 1. That is 1 mole of hydrogen is required to convert 1 mole of unsaturated compound into which compound? Saturated compound. How is hydrogenation is better process? This degree of unsaturation is telling that here what is present? Unsaturation is present. Hydrogenation is a process. Addition of hydrogen to any unsaturated compound is called as hydrogenation reaction. But here we are trying to find what is degree of unsaturation, that how many moles of hydrogen it is short from saturated compound. Okay. Next example if we take, let us take C2H2. C2H2 is ethyne, and what is structure? HC triple bond CH. So what is present here? Triple bond is present. Now this is unsaturated compound. If we add hydrogen, first we are writing it saturated compound. So saturated compound for 2 carbon atom is ethane, C2H6. Now if we balance this reaction, then how many moles of hydrogen are required? 2 mole. So if we take 1 mole of ethyne, then how many mole of hydrogen are required? From stoichiometry we can see 2 mole of hydrogen are required. For 1 mole of C2H2, how many moles of hydrogen required? 2 moles. So what we can say, degree of unsaturation of C2H2 is how much? 2. Clear? 1 mole of unsaturated compound is converted to saturated. How many moles of hydrogen are required? 2 moles. So degree of unsaturation for C2H2 is how much? 2. Clear? Now next, this is open chain compound. If there is any cyclic compound, then what is degree of unsaturation? We have to convert that cyclic compound into open chain compound. So if we take this compound, students, you have to tell first what is name of this compound. I have written a cyclic structure. You have to tell what is IUPAC name of this compound. 4 carbon atoms are given. Degree means mole. No, degree is different. Mole is counting unit. So what is name? Very good. Name is cyclobutane. Very good. What is name? Cyclobutane. Students, can you tell molecular formula for this compound? Now you have to tell what is molecular formula for cyclobutane. That how many carbon atoms are present, how many hydrogen are present. What is molecular formula for cyclobutane? C2H4. How it is possible? Butane, total 4 carbon atoms are present. If you find out molecular formula, what you get? Here what is present? Carbon and 2 hydrogen, CH2. CH2. Here CH2, CH2. Carbon has total 4 valencies. So 2 carbon single bond. So how many hydrogen are present? 2 hydrogen. So what is molecular formula for this compound? It is C4H8. Remember, this is not butane. Butane is C4H10 and cyclobutane is C4H8. Now if we add hydrogen and we have to convert this into open chain compound. So open chain compound of cyclobutane will be butane. So how to write structure? CH3, CH2, CH2, CH3. And what is molecular formula for butane? C4H10. So if you check, C4H8 plus H2 gives C4H10. So already reaction is balanced. So how many mole of hydrogen are required? If we take 1 mole of cyclobutane, then 1 mole of hydrogen is required. So what we can say here, degree of unsaturation of C4H8, here it is how much? 1. Clear? So 3 points we have written. First is conversion of unsaturated double bond to single bond, then triple bond to single bond, then we have written cyclic compound to open chain compound. So from here what conclusion we get? That if a compound contain only one single bond, then what is degree of unsaturation? 1. If a compound contains only one triple bond, then what is degree of unsaturation? Degree of unsaturation is 2. Okay. Next is if a compound is cyclic. If a ring is present, then what is degree of unsaturation? Degree of unsaturation is 1. So this is very important conclusion. What we have to remember here, that if a compound contains only one double bond, this is representation for double bond, then for double bond what is degree of unsaturation? 1. If a compound contains one triple bond, what is degree of unsaturation? 2. If a compound contains one ring, then what is degree of unsaturation for this? Degree of unsaturation is 1. So this points we have to remember, that for double bond degree of unsaturation is 1, for triple bond degree of unsaturation is 2, and for one ring degree of unsaturation is 1. Now I am asking one question. You have to tell degree of unsaturation of benzene. What is degree of unsaturation of benzene? Try to solve this. For that you should know structure of the benzene. For 2 triple bond degree of unsaturation, what you are saying? 6. How it is possible? For 2 triple bond degree of unsaturation is 4. For benzene, what is degree of unsaturation? Students are saying 1, 3. Some students are saying 4. 4 is correct. How to draw structure of benzene? This structure is benzene. What is molecular formula for benzene? C6H6. What is molecular formula? C6H6. Now, if you check in this compound, it contains one ring is present, 6-membered ring. And how many double bonds are present? 3 double bonds are present. Now, for one ring, what is degree of unsaturation? 1. And for 3 double bond, what is degree of unsaturation? 3. So, what is total degree of unsaturation of benzene is how much? 1 plus 3, 4. Here, this will be the answer. Clear? So, it is very easy. For benzene, if you know the structure C6H6, easily we can find degree of unsaturation. Now, next is, if we do not know structure of benzene, only we know molecular formula, then we can also calculate degree of unsaturation. So, if we write degree of unsaturation from molecular formula of compound, then also we can find out what is degree of unsaturation. So, what is formula for this? Degree of unsaturation is number of carbon atom plus 1 minus number of hydrogen atom plus halogen atom minus nitrogen divided by 2. So, this is formula for degree of unsaturation if molecular formula is given. What is C here? C is number of carbon atoms. Here, what is H here? It is number of hydrogen atoms. In molecular formula, what is X here? X represent halogen. Number of halogen atom. Halogens, you know, fluorine, chlorine, bromine, iodine, these are called as halogens. And N represent number of nitrogen atoms. Okay. So, whenever molecular formula is given, what is formula for degree of unsaturation? C plus 1 minus H plus X minus N divided by 2. Okay. Write down this formula in your notebook. Then we'll apply some, I will give some molecular formulas. You have to tell what is degree of unsaturation by applying this formula. Okay. So, this is very important formula. Number of carbon atom plus 1 minus hydrogen plus halogen minus nitrogen divided by 2. Okay, so, if formula is given, then we have to apply this. If structure is given, directly we can apply these to calculate degree of unsaturation. Okay. Now, question is, find degree of unsaturation for following compounds. So, first is C2H6, C2H4, C2H2, C3H6O. Next is C5, H13N, next is C5, H11NO, next is C6H6. So, these molecular formulas are given, and what we have to find, what is degree of unsaturation, but we have to apply formula for this. Now, structure is not given. What is given? Formula is given. First, try to solve, take 1 or 2 minutes. After 2 minutes, we'll discuss all these 7 and send all answers collectively from 1 to 7. In one single message, you have to write all answers. Do not send individual message. Try to solve this. Simple formula you have to apply. I am writing formula here again. C plus 1 minus H plus X minus N divided by 2. So this formula you have to apply. College reopen date. You will get notification. So let us start from first part. Tell answer for number 1. What is degree of unsaturation? C2H6. C2H6 1 0 correct. 0 is answer. If you apply here in C2H6, how many carbon atoms are present? 2. So 2 plus 1 3 minus. How many hydrogen atoms are present? 6. So 6 divided by 2. So 3 minus 3. What is degree of unsaturation? 0. Reason is because C2H6 is a type of which compound? Saturated compound. There is no degree of unsaturation. So that's why degree of unsaturation is 0. Now let us solve second part. Tell degree of unsaturation for C2H4. For C2H4, what is degree of unsaturation? For C2H4, what is degree of unsaturation? 1. Very good. How many carbon atoms are present? 2. So 2 plus 1, 3. How many hydrogen? 4. So 4 divided by 2. So 3 minus 2, how much degree of unsaturation? 1. Clear? Let us move to third part. Tell for third, what is degree of unsaturation? C2H2. For C2H2, what is degree of unsaturation? Very good. Students are telling 2. But how many carbon atom? 2. So 2 plus 1, 3. How many hydrogen? 2 hydrogen. So 2 divided by 2. 2 by 2, 1. 3 minus 1. What is degree of unsaturation? 2. Okay. Now let us move to fourth part. Tell answer for fourth. C3H6O. What is degree of unsaturation for C3H6O? Some students are saying 2, 0, 2, 1. So first tell, how many carbon atoms are present? 3 carbon atoms. So 3 plus 1, 4 minus. Now there is no role of oxygen atom in degree of unsaturation. Only hydrogen, halogen and nitrogen. So leave this oxygen. How many hydrogen are present? 6. So 6 divided by 2. So 6 by 2, 3. 4 minus 3. What is degree of unsaturation is 1. Here, so degree of unsaturation for C3H6O, how much it is? 1. Now tell answer for fifth part. C5H13N, what is degree of unsaturation? Next fifth part, C5H13N. Ratna, try again. You are getting 1. No, answer will be 0 for this. How much degree of unsaturation will be there? 0. Because now nitrogen will participate. Now 5 plus 1. C5H11NH2. So 5 plus 1, 6. Now what we have to write? Hydrogen plus halogen minus nitrogen. So hydrogen are 13, nitrogen is 1. 13 minus 1, how much? 12. So 12 divided by 2. So it will 6 minus 6. What is answer? 0. Clear? So what is formula? Hydrogen plus halogen minus nitrogen. So number of nitrogen atom we have to minus. 13 minus 1, 12. So 12 by 2, 6. 6 minus 6 is 0. Okay. Now tell answer for sixth part. C5H11NO. Sixth part. What is degree of unsaturation? Students are telling 1. 1 is correct answer. Now, oxygen will not participate. Only 5. Carbon atom plus 1. 5 plus 1, 6. Minus. Hydrogen minus nitrogen. So, 11 minus 1 is 10. 10 divided by 2. So, 6 minus 5. What is degree of unsaturation is 1. Here. Now next part, C6H6. How many carbon atoms? 6. 6 plus 1, 7. Minus 6 divided by 2, 3. So 7 minus 3, what is degree of unsaturation? 4. Okay. And if you check these structures, that is ethane, ethene, ethyne and benzene. These examples we have done from structure also. So this is first type of question where molecular formula is given and what we have to find degree of unsaturation. Okay. Now second type of question where structure is given and what we have to find degree of unsaturation we have to find. So maximum times they will give, if they only give question on degree of unsaturation, they will draw a structure and then they will ask what is degree of unsaturation. So let me write a question on that. Find degree of unsaturation, or it is called as double bond equivalent, or it is called as hydrogen deficiency index for following compounds. First part is, now remember this is representation of benzene. In benzene, they will not show three double bonds. They will write this circle. So this is benzene, and what is attached here? COOH. Second structure is next structure. So first solve these three, then we'll go to next. So these are important when structure is given, how to find degree of unsaturation. Try to solve. Then I will write some other examples also. First, you have to solve these three. Start from number 1. First, tell what is degree of unsaturation. So let us do one by one. And first, you have to tell answer for first part. Students are saying degree of unsaturation is 4. Now let us, if I draw this structure again, then you have to tell. I will write expanded structure. Now in benzene, this is present. Now on benzene, C double bond OH is present. This is called as benzoic acid. So what is structure of our benzoic acid? C double bond O, OH. Now count degree of unsaturation for this. Now easily you can find degree of unsaturation. So first, degree of unsaturation, how many rings are present? One ring is present. For ring, degree of unsaturation 1. How many double bonds are present? 1, 2, 3, 4. So degree of unsaturation 4. 4 plus 1, what is degree of unsaturation? 5. So this will be the answer. So you have to draw expanded structure so that you can know how many double bonds and how many rings are present. So in this structure, one ring and total how many double bonds? 4. That's why answer is 5. Okay. Now tell me answer for second part. In second, how many double bonds, how many ring? You have to tell what is degree of unsaturation for second part. Sairaj 7. Let us take another. Tell students, all students have to answer. What is answer for this? Many students are saying 7. Very good. So what is present here? First ring, that is benzene. Let us draw benzene. Next simple, it is cyclohexane. Next 4-membered ring. And what is present? Double bond. Oxygen. Now what is degree of unsaturation for this? So if we count, first how many rings are present? 1, 2, 3. 3 rings are present. For one ring, degree of unsaturation 1. For 3 rings, degree of unsaturation is 3. Now next check how many double bonds are present. 1, 2, 3, 4. So 3 plus 4, what is degree of unsaturation? 7. Clear? Now tell answer for third part. What is degree of unsaturation for third? For second answer is correct 7. Now solve for third part. What is degree of unsaturation? How 5 is possible? 5 is not possible. Now first check how many rings are present. So I will highlight this. This is first ring. Can you check? This is a first ring. Now this is second ring. These type of compounds are called as bicyclo compounds. So it contains total how many rings are present? 2 rings are present. So in this question if I write how many rings are present? 2 rings. How many double bond are present? Only 1 double bond is present. So now what is total degree of unsaturation? Degree of unsaturation will be 2 plus 1, it is 3. Clear? Can you find degree of unsaturation? It is easy to find. Any doubt in these 3 parts? First, second, third? Then I will write 2 or 3 more structures where you have to tell degree of unsaturation. Clear? So let us move to next 3 part I will give again. You have to solve. Next fourth is now expanded structure you have to draw. These are typical type of questions I am giving you. Now solve these two. You have to tell what is degree of unsaturation in these 2 compounds. For first answer you are telling 6. Try to draw expanded structure, then you will get degree of unsaturation. Many students, some students are saying 6, 9, 8, many answers we are getting. 5 also. So let us do one thing. I will draw expanded structure, then you will tell what is degree of unsaturation. First ring. Second is ring where what is present? Benzene. Here cyanide. Cyanide is C triple bond N. COCH3, C double bond O, CH3. OH is oxygen single bond hydrogen. C double bond O, OH, oxygen single bond hydrogen. Now count degree of unsaturation. Then you will get correct answer. Now find total degree of unsaturation for this compound. First identify how many rings are present here. 1 plus 1, 2 rings are present. For 2 rings, degree of unsaturation 2. Next you have to count how many double bonds are present. 1, 2, 3, 4, 5. 5 double bonds plus 5. Now how many triple bond are present? 1. For 1 triple bond, what is degree of unsaturation? 2. So what you get answer is 9. Clear? So if you know these functional groups, you can draw expanded structure. Then easily what we can find degree of unsaturation. Now important, in exam they will not give this expanded structure. First structure they will write. You have to write expanded structure and then find degree of unsaturation. Clear? Now move to next 5th part. Tell what is degree of unsaturation for 5th. For 5th part answer is 6. Let us check degree of unsaturation. Now no need to draw structure. This is first ring. This is second ring. So total 2 rings are present here. So for ring, what is degree of unsaturation? 1 plus 1, 2 ring. Clear? How many double bond are present? 1 plus 1, 2 double bond are present. How many triple bond are present? 1 triple bond. For triple bond, what is degree of unsaturation? 2. So what is answer for this? Degree of unsaturation is 6. Here. So any doubt in these questions? If molecular formula is given, if structure is given, then how to find degree of unsaturation? Well students, do you understand this complete part? How to find degree of unsaturation? Okay, let us move to next part. Now this is required in first question. This is simple, degree of unsaturation calculate. Now next application in isomers, if we have to molecular formula is given and we have to predict how many possible structures are there. So what is significance of this degree of unsaturation for making isomers? So what we write, significance of degree of unsaturation. So first point is, if there is a molecular formula is given and its degree of unsaturation comes out to be 1, then what we can think from here, that this compound contains either one double bond or it contains one ring. So what we can say in this compound, what are possible things? Either one double bond is present or either one ring is present. So first we draw all the possible structure where double bond is present, then we draw all the possible structure where one ring is present. Ring can be of 3 carbon, 4 carbon, 5 carbon, 6 carbon. That depends on what is molecular formula given. Clear? Now next is, if degree of unsaturation is given 2. Now what are possible cases for degree of unsaturation 2? First case is either one triple bond is present, because for triple bond, how many degree of unsaturation? 2. Next case is that in that compound, how many double bond are present? 2 double bonds are present. This is also possible. Next case is compound contains 2 ring. This is also possible. And next case is compound contain 1 double bond and 1 ring. This is also possible case. Clear? So these all are possible cases for degree of unsaturation 2. What is possible? Either 1 triple bond, either 2 double bond, either 2 ring, or 1 double bond plus 1 ring. So these are possible cases for degree of unsaturation 2. Next, if we write possible case for degree of unsaturation 3. So let us write from degree of unsaturation 3. First I am writing what are present? 3 double bond are present. Next I am writing 3 ring are present. Possible. Next we make combination with triple bond. So if 1 triple bond is present, how much degree of unsaturation is consumed? 2. 1 is left. So what is present? 1 double bond. Next thing, what is possible? 1 triple bond plus 1 ring is possible. Next possible case is 2 double bond and 1 ring is possible. And next possible case is 1 double bond and 2 ring. So these are possible cases for degree of unsaturation 3. But remember, degree of unsaturation 3 they will not give in questions. Maximum in NEET exam will get degree of unsaturation 1 and 2. They will ask how many isomers are possible for any compound. So maximum time they will give molecular formula where degree of unsaturation 1 or degree of unsaturation 2. 3 is very less chances, because if you can check, many possible combinations are there. So many structures are possible. So they will not ask about these things, degree of unsaturation 3, maximum 1 and 2. Clear? Now important is, I will give a molecular formula. Then you have to find out how many possible structures are there for that compound by using this degree of unsaturation. So remember this degree of unsaturation 1 and 2. So question is, draw all possible structures of C4H8. Very easy question this is. Not asking tough question. Simple, only 4 carbon atoms, C4H8. How many possible structures are there for this compound? Try to solve. Try to draw structures. First, find out what is degree of unsaturation. First, tell me 16 structure. How 16 structures are possible? 16 are never possible. First, if we calculate degree of unsaturation, easily you can find degree of unsaturation. What is degree of unsaturation? How many carbon atoms are present? 4. So 4 plus 1, that is 5, minus 8 divided by 2. So what is degree of unsaturation? 1. Now from degree of unsaturation 1, what we can assume, either one double bond is present or either one ring is present in the compound. Okay. So let us first draw the structures in which one double bond is present. So first I am drawing the structure. 4 carbon atoms are present in straight chain. Double bond is present at which position? First position. What is name of this compound? But1ene. Now it is also possible double bond is at second carbon. So what is name of second compound? But2ene. Next possible case is if we take 3 carbon atoms in straight chain, 1 methyl branching, and at one position what is present? Double bond. So it is 2 methyl propene. So 3 structures are possible for double bond. 4 carbon atoms in straight chain, double bond at first position, double bond at second position, 3 carbon atom and 1 double bond is present. Clear? Next, if we take one ring, so ring means 4 carbon atom ring is possible, or either 3 carbon atom ring and 1 methyl is present. Because ring can be also, sir, can we think number of structure 2 into du? No, not possible. There is no general conclusion. You have to draw all possible structure. There is no formula for this. So total how many structures we get? Open chain structure 3, that is alkenes, and 2 are cyclic compounds. So how many total structures are possible? So total answer is 5. From degree of unsaturation just only helps you that how to find that double bond is present, triple bond is present, how many double bond, how many triple bond, how many rings, but possible structures you have to draw by yourself. Is there any easy method to identify structures from structure? It is very easy. You have to draw expanded structure. We have done 5 examples on that. Clear? So this is possible structure of C4H8. Now I am giving you 2 questions which you have to attempt after the class. In next class we will discuss those 2 examples. Okay. So without taking any help, you have to draw by yourself. Draw possible structures, all possible structures of C5H10. And next is draw all possible structures of C4H6. So these 2 questions are your work. After the classes, you have to apply this. Just remember, from degree of unsaturation you will get an idea. After that you have to draw the structures. Okay. So in next class we will discuss these 2 questions and next move to next topic, that is basics of structural isomerism. Okay. So students, thank you for today. We will meet in next class with isomerism topic.
Hi students, hello, good morning. Good morning, Ra. Today's botany topic is nucleic acids. Nucleic acids. The knowledge about the nucleic acid is very, very essential to understand two very important topics in secondary intermediate, such as biotechnology. Another one is the molecular biology. To get sound knowledge in such two chapters, the basics of the nucleic acids is the very, very essential and mandatory. So concentrate very well to get basic concepts, to enjoy in such higher topics in higher classes. Nucleic acids. DNA and RNAs are very good example for nucleic acids. Is it macromolecule or micromolecule? Nucleic acid is a macro or micro? Yes, macromolecules, because their molecular weight is greater than 1000 daltons. Greater than 1000 daltons molecular weight is. And it is the polymer of Na, full form nucleic acid. It is the polymer of nucleotides. Polymer of nucleotides. The building blocks of nucleic acids are nucleotides. Yes, macro. What are the building blocks of the protein? What are the building blocks of a protein molecule? Protein is a macromolecule. Amino acids are the building blocks of the protein. Sugars are the building blocks of polysaccharides. Yes, likewise nucleotides. Nucleotides are the building blocks, are structural units of nucleic acids. Okay, these are the nucleic acids are two types. Based on type of sugar molecule is present in nucleic acid, these are two types. If the type of sugar is the deoxyribo, then that is a DNA. Whereas in RNA molecule, the sugar molecule type is the ribonucleic acid. Ribo sugar, here deoxyribo sugars are there. The basic or major variation between DNA and RNA is the type of sugar. The type of sugar in DNA is deoxyribo sugar, whereas in RNA, type of sugar is the ribose. Okay. It's the DNA double stranded structure, but RNA is the single stranded structure. RNA is single stranded, only one strand is there, whereas in DNA, two strands. Hence, we can say it is a double stranded, this is the single stranded. Observe here, nucleic acids are the building block, are the building blocks of genetic material. Genetic material is made up of nucleic acids. Genetic material is made up of nucleic acids. Nucleic acids are made up of nucleotides. Nucleotides. A little bit confusion there here, be alert. Nucleotides are the building blocks of nucleic acids, but nucleic acids are the building blocks of genetic material. Okay, na? It means genetic material is made up of DNA, RNA. DNA and RNAs are made up of nucleotides. In most of the organisms, 99% of the living organisms, DNA is the only genetic material. Whereas in very few viruses only, RNA is the genetic material. Actually, viruses are non-living. These are non-living things. Except few, not all, except some viruses, remaining every living thing in this universe, genetic material is the DNA. Okay, but most common functioning of RNA is carrying of genetic message from DNA to required places. It is the messenger, hence it is RNA is called as the messenger. It is not a true genetic material RNA. RNA acts as a just information carrier. The information which is present in DNA molecule carried to cytoplasm from nucleus. Generally, DNA is found inside of the nucleus. For example, it is one plant cell. In that nucleus is there. The major location of DNA molecule is the nucleus. The information is required to synthesize some macromolecules present on DNA. The DNA is copied into one single stranded RNA molecule. That RNA transferred to the cytoplasm for the functioning. So, information transferred from DNA to cytoplasm in the form of a Xerox copy, that is the RNA. Hence, another name of the RNA is the messenger RNA. Another name of RNA is functionally it is called messenger or message carrier. From where to where it carries message? Nucleus to cytoplasm. Yes, somebody asking Mr. Venkat Nageswar Rao, your question is which virus consisting of both the DNA and RNA? In this universe, there is no single virus consisting of both DNA and RNA. Either DNA or RNA are present in viruses. No scope for presence of both the DNA and RNA molecules. For example, in coronavirus, HIV virus, influenza virus, RNA is there. Genetic material is the RNA. In all plant virus also, genetic material is RNA. Best example for plant virus, tobacco mosaic virus. Tobacco mosaic virus. Are you getting? There is no virus in this universe that possesses both DNA, RNA at a time. Either DNA or RNA only present. Either DNA or completely RNA. RNA is genetic material in most of the plant viruses and some of the animal viruses like coronavirus. In RNA is there. Later, by the one mechanism of transcription, RNA is involved in formation of DNA molecule. That story is different. You can learn in higher classes. Okay, is it clear? Mr. Venkat Nageswar Rao. Now, elementary knowledge about the nucleic acids. Like protein molecule, we learned in last class. Nucleic acid is also heteropolymer. Polymer, combination of many molecules. Hetero, all these molecules are not identical to each other, differ. Hetero means different. Different types of building blocks involved in construction of DNA molecule. Hence, both DNA and RNA are example for hetero. Every protein is a hetero. But polysaccharide, two types are there, homopolysaccharides as well as heteropolysaccharides. But every protein molecule, every nucleic acid is an example for heteronucleotides. Okay. Another name of this nucleic acid is the polynucleotide. Many nucleotides linked with another to form DNA. Another name of that is polynucleotide. Like protein also called as polypeptide. Other name of the protein is a polypeptide. Likewise, another name of DNA, RNA is a polynucleotide because each DNA or RNA is made up of many nucleotides. Okay. Observe here in this image. This is the one nucleotide. It is the second nucleotide. Nucleotide number 3. Nucleotide number 4. Likewise, the lakhs of nucleotides are joined with another one, linked with another one to form a long chain. The name of the chain is, name of this polymer is polynucleotide. Even in protein molecule also many amino acids are linked with another one to form a long chain of amino acids. Issue. Yes, yes. Virus is a connecting link between both living things and non-living things. Virus is not a completely living, not like completely non-living. When it is outside of the host, non-living. When it is inside of the host, that may be either plant cell or animal cell, then only that can exhibit some kind of living properties. Okay, yes. Okay, clear? Now, lesson this 10 minutes class is the very, very essential. Students, concentrate. 10 minutes class is the very, very essential. Because the basic or fundamental things about the DNA and RNA. A DNA molecule is made up of several nucleotides. Every nucleotide is made up of 3 structural units. One is the nitrogen base, second one is the sugar molecule, third one is the phosphate. One question ready here. How many constituents are found in a nucleotide? How many number of constituents are there? 3. Nitrogen base, sugar molecule, phosphate. First of all, you have to learn about the sugar molecule. What kind of sugars are found in DNA? Or nucleic acids. Chemical composition of nucleotides. Every nucleotide is made up of 3 chemical constituents. One is the sugar, second phosphate group, another one is the nitrogen bases, short name NB. Nitrogen bases. We can use the simple word base also. Base nothing but a nitrogen base. What kind of sugar is found in DNA and RNA? Based on number of carbon, the sugar is the pentose. 5 carbon contain. 5 carbon contained sugar molecule is the pentose. With the 5 number of carbons are 5 carbons are there. It is the sugar molecule. 5 membered ring. That this is the first carbon, second carbon position, third carbon, fourth one, fifth carbon is found outside of the cycle. It is the 5th carbon. But his oxygen is there, oxygen atom. Total number of carbons are 5, but within a circle, the number of carbons are 4 only. Pentose sugar. As you know, each carbon possesses 4 valency positions. One position, this bond, second position, third and fourth position, hydrogen, hydroxy group OH. Even in second carbon also, hydrogen OH. Third carbon, hydrogen OH. At fourth carbon, only hydrogen atom is there. Only hydrogen atom. Fifth carbon, CH2OH group is there. Now, how many number of carbons are there in the entire sugar molecule? Count 1, 2, 3, 4, 5. In formula C5. How many number of hydrogens are there? Count the number of total hydrogen atoms in this molecular structure. Hydrogen 1, 2, 3, 4, 5, 6, 7, 8 plus 2. 10H10. Okay. Likewise, count the total number of oxygen atoms in this sugar molecule. Total number of oxygen atoms. Number of carbons 5. Number of hydrogens 10. I am asking about number of oxygen atoms. Oxygen atoms. Oxygen 1, 2, 3, 4, 5, O5. O5. So, it is the formula for ribose sugar. The sugar ribose is present in DNA or RNA? Sugar of RNA molecule. It is the sugar of RNA. What is the difference between RNA sugar to DNA sugar. What is the difference between sugar molecule of RNA to DNA? Almost all both the sugar molecules are similar. Yet only one difference is there at one place. This is the deoxyribose sugar. Its formula C5H10 similar, but O4 is there instead of 5. Identify difference between sugar molecule in RNA, that is the ribose, and sugar molecule in DNA is the deoxyribose. Formula C5H10O5, whereas in deoxyribose sugar molecule formula is the C5H10O4. The difference clearly visible at second carbon. In second carbon, just hydrogen atom is found instead of OH group. Identify the variation here. At the second carbon, apart from hydrogen atom, another valency position is OH. Whereas here, both the valency positions are H and H. What is the difference between OH group to the H group? Hydrogen atom is the deficit. That's why it is the O4, it is the O5. Identify here, D for deficiency of one oxygen atom in a ribose. Then the name of that ribose is the deoxyribose. Okay, are you getting this idea? How are you going to differentiate the deoxyribose sugar from the ribose molecule? When compared to the formula of the ribose molecule, one oxygen atom is the deficit. Hence, the ribose is named as deoxyribose. Okay. Such kind of difference is found in the second carbon only. In ribose sugar, OH group is found at the second carbon, whereas in deoxyribose sugar, just hydrogen atom is found at the second carbon. With the major variation. If the sugar is the ribose, that molecule is the RNA. If the sugar is the deoxyribose, then the nucleic acid is called as a DNA. DNA, first letter D for deoxyribose sugar contain nucleic acid. In RNA, first letter R for ribose contain nucleic acid. Okay, is it clear? Shortcut diagram of this deoxyribose or ribose sugar molecule. Shortcut diagram. This is the carbon 1, 2, 3, 4, 5th carbon. Once recollect or remember how many chemical constituents are there in a nucleotide. Sugar is there, phosphate, nitrogen base. Discussion about the sugar completed. Information about the sugar molecule is over. Next constituent is the phosphate group. Another one is the nitrogen base. First of all, you have to learn about nitrogen bases. So later we will go to phosphate area. Yes, 3 constituents. Nitrogen bases usually bound to sugar molecule at the first carbon only. NB. What is the full form of NB? Nitrogen base. Nitrogen base always linked to first carbon of sugar only. The bond present between both sugar and nitrogen base is glycosidic bond. Glycosidic bond. Already you learn about this kind of bond in polysaccharides. In polysaccharides, to adjacent sugar molecules linked by the glycosidic bonds. In cellulose is the polymer of glucose, starch polymer of glucose, glycogen polymer of glucose, inulin is the polymer of fructose molecules. Such as the glucoses or fructoses are linked with another one by forming glycosidic bond. So glycosidic bond not only found in polysaccharides, also found in nucleic acids. Okay, the bond present between sugar molecule and nitrogen base is the glycosidic bond. Now these 2 constituents together called as These two together called nucleoside. Nucleoside. Now the nucleoside plus phosphate. Third constituent of the nucleotide is the phosphate. It is the fifth carbon of sugar molecule. Fifth carbon of sugar molecule. Phosphate linked to fifth carbon of sugar. The name of this bond is the phosphoester bond. Phosphoester bond. Phosphate bind to fifth carbon of sugar by the phosphoester bond. Nitrogen base linked to first carbon of sugar by the glycosidic bond. So two types of bonds are essential to construct overall nucleotide structure. Sugar plus nitrogen base is the nucleoside. Nucleoside plus phosphate is the nucleotide. Some chances are there to confuse between nucleoside and nucleotide. If the letter is a T, if the letter is a S, side different from tide. Try to remember this one. Tell me, nucleotide is the bigger molecule or nucleoside is the bigger molecule? Tide is bigger, side is bigger. Nucleotide is always bigger than that of nucleoside. Tell me, what is the chemical compound present in nucleotide, not in nucleoside? Name the chemical compound. Yes, right answer, good. Chemical compound present in nucleotide, not in nucleoside. Yes, phosphate. Phosphate is the right answer. What are the chemical constituents are common in both nucleotide and nucleoside? Name the chemical constituents are common in both nucleotide and nucleoside. Sugar and nitrogen base. Sugar and nitrogen base are common in both. One more question. Name the bond present in nucleotide, not in nucleoside. Chemical compound is the phosphate, present in tide only, not inside. Likewise, I am asking one more kind of question. Name a bond absent in nucleoside but present in nucleotide. Yes, phosphoester bond. Glycosidic and phosphoester bonds are found in nucleotide only, whereas in nucleoside only glycosidic bond is there. So it is the reason to say nucleotide is the bigger than nucleoside. Once again, I will give the definition. Base plus sugar is nucleoside. Base plus sugar plus phosphate is nucleotide. Or another meaning for the nucleotide is nucleoside plus phosphate with phosphoester bonds is a nucleotide. This kind of nucleotides are several or millions are polymerized to form DNA or RNA. If the sugar in this nucleotide is the ribose sugar, that entire macromolecule is RNA. If the sugar of this nucleotide is the deoxyribose sugar, the entire polymer is called as DNA. It is one of the basic variation between DNA to RNA molecules. Okay. It is the structure of nucleotide. In case of nucleotides, only these two constituents are there. Tell me once again, what is the name of this bond? The name of this bond. Try to give answer. Yes, good. Glycosidic bond. Glycosidic. N-glycosidic. And the bond present between fifth carbon of a sugar. This is the fifth carbon, it is the fourth carbon, third, second, first carbon. Name of this bond. Ester bond. Yes. Ester. Phosphoester. Two types of bonds. Structure of polynucleotide chain. Single nucleotide is the monomer or biomicromolecule, but polynucleotide chain is macromolecule. Nitrogenous base is linked to pentose sugar through the N-glycosidic bond, just completed recently. Both together called as the nucleoside. Nucleoside. If the phosphate is also binded to it, that becomes as a nucleotide. Phosphate to the fifth carbon. It is the ester bond. Nucleotide. Now next topic is types of nitrogen bases. Types of nitrogen bases. Broadly classified into two categories: purines, another category is the pyrimidines. Purines and pyrimidines. Both adenine and guanine are example for purines. Cytosine, thymine, uracil are example for pyrimidines. Example for purines: adenine, guanine. Pyrimidines: cytosine, thymine, uracil. Short name of adenine is A, guanine G, C for cytosine, T for thymine, U for uracil. U for uracil. Try to remember the names of these 5 types of nitrogen bases. A for adenine, G for guanine, C cytosine, T thymine, U uracil. A, G are example for purines. C, T, U are example for pyrimidines. Both the purines, adenine, guanine, found in all DNA molecules. In case of pyrimidines, just C and T are present, U absent in DNA. So types of nitrogen bases found in DNA. These first 4, A plus G and C plus T, are found in DNA molecule. Whereas in RNA, except thymine, remaining all are present. In RNA, adenine, guanine, cytosine and uracil is present instead of thymine. There is no single nucleic acid that possesses both the thymine and uracil at a time. In out of these two, only one present in every nucleic acid. In RNA, A, G, C, U are present. Whereas in DNA, A, G, C, T are present. So adenine, guanine, cytosine are common in all nucleic acids. Thymine found in DNA only, uracil found in RNA only. It is the second difference. Already you might have learned about the sugar molecules present in DNA, RNA. In DNA, sugar is the deoxyribose sugar, RNA ribose sugar. Second important difference is type of pyrimidine. Thymine found DNA only, uracil found in RNA only. Remaining 3 types of nitrogen bases are common in both DNA and RNA. Adenine, guanine, cytosine, commonly found in both DNA molecules and RNA molecules. And another important aspect, observe here, the chemical structures of purines and pyrimidines. Chemical structures of purines and pyrimidines. In both adenine and guanine, 2 rings are present, dicyclic structures. Here also green color structure, 2 rings are there. So both adenine, guanines are dicyclic structures. Whereas the 3 types of pyrimidines, monocyclic structures, single CN rings are there. Single ring, single ring, single ring. So these 3 are monocyclic, these 2 are dicyclic. Adenine, guanine means purines are dicyclic structures, pyrimidines are monocyclic structures. These are dicyclic structures or double ring structures, single ring or single mono ring cyclic structures. is one of the major structural variation between purine to pyrimidine. Purines are only 2 types, but pyrimidines are 3 types. All types of purines are found in both DNA and RNA. In case of pyrimidines, in DNA, C and T are present, whereas in RNA, C and uracilone is there, thymine is absent. From these types of nitrogen bases, we can ask some questions here. Tell me, name the nitrogen base that never found in RNA molecule. What is the nitrogen base? Absent in RNA but present in DNA. Thymine. Thymine, T. Name the nitrogen base absent in DNA but present in RNA. Not in DNA. Yes. Uracil. Likewise, one more question. Differences between purine bases to pyrimidine bases. Difference between purine to pyrimidine. We can see the structural variation between purine to pyrimidines. Double cyclic structures or dicyclic structures, but pyrimidines are single cyclic structures or monocyclic structures. Polynucleotides are the polymer of nucleotides. DNA, RNA are polynucleotides. Observe here, it is a long chain of nucleotides and that is called polynucleotide. Observe in each and every nucleotide, one is the base, second one is the sugar molecule, third phosphate. Here also base, sugar, phosphate, nitrogen base, sugar, phosphate, nitrogen base, sugar, phosphate. Identify, B, concentrate. Try to give the proper answer for my question. Identify the difference from one nucleotide to another nucleotide. You take any 2 nucleotides. Guess what kind of variation is there? What is the difference from one nucleotide molecule to second nucleotide molecule? Have you identified any kind of variation? Yes, good. NB, nitrogen base. For example, here, nitrogen base is the T for. What is the full form of the T? Yes, good. Bases, okay. What is the full form of T? T for thymine. A for. Give the answer without seeing textbooks or notes. Search in your brain. A for adenine. Yes. T for thymine. G for full form of the G. Yes, guanine. Last one C. C for cytosine. Yes, good. C cytosine, G guanine, T thymine, A for adenine. Now, on this basic information, you can guess it is the polymer of DNA molecule or RNA molecule. Is it DNA or RNA? This molecule is a DNA or RNA? Yes, good. How can you say it is not RNA, it is only DNA? Good. Your answer is right, exactly, perfectly right. How can you say? Yes, to say it is a DNA, not RNA, thymine is there. Thymine found in DNA. Uracil is absent. That is also, you can say it is a DNA molecule. Yes, good, nice. And another important concept. How can you say it is a sugar is the deoxyribose? This is the sugar is a deoxyribose only. Deoxyribose sugar. There is no any kind of formula to identify it is a deoxyribose or ribose. But one logic is there. You can apply a logic. Students, apply a logic to identify if this is the sugar is not a ribose, it is only deoxyribose. What kind of logic behind here? No, how can you identify here? O2 is there, O2 not there, there is no any kind of formula here. That is not a clear logic. No. My question is, how can you say it is a deoxyribose? It is not a ribose. According to this diagram. No, structural evidence is required. Without that evidence, I can say yes. Cyrus, excellent, very good. Thymine always bind to present in DNA only. In DNA, sugar is deoxyribose. Yes, Cyrus, good. No, Vaishnavi, that is not a correct logic. Yes, Viber, good. Viber, is it a real name? Alias, I think. Yes, thymine is a specific kind of nucleotide found in DNA only. In DNA, sugar is deoxyribose sugar. Thymine never bind to ribose. Uracil never bind to deoxyribose. Lack of uracil, presence of thymine, we can easily guess it is a deoxyribose sugar and this molecule is the DNA. It is the logical evidence. Yes, Madhavi, good. Farine, good. Next concepts. Answer for this question. Not a polymer from following. Yes, good. Second option. Protein is the polymer of amino acids. That's why not a answer. Not a polymer. Starch is the polymer of glucose molecules. Cellulose is also polymer of glucose. But purely nucleotide is the micromolecule or it is the building block of another polymer like DNA. Nucleotide. Good. What is the nucleotide? Nucleotide. Answer. Yes. Fourth option. Sugar plus base nucleoside plus phosphate nucleotide. Sugar plus nitrogen base nucleoside. Okay, nice. Next question. What is the pyrimidine that is the common in both DNA and RNA? Pyrimidine, not purine. What is the pyrimidine common in both the nucleic acids, DNA and RNA? Is it that common one is the cytosine or uracil or thymine or adenine or guanine? Yes, cytosine. Present in both the DNA and RNA. What about adenine? Purine common in both the DNA and guanine also common purine both DNA RNA. Thymine is not common. Peculiarly, it is found in DNA only. Okay, cytosine is the common type of pyrimidine. It can satisfy these two words, pyrimidine word and DNA RNA. Both DNA and RNA are example for nucleic acids. Why these are the DNA and RNAs are called as acids? Is there any logic is there to say DNA, RNAs are nucleic acids? The acidic nature of this DNA, RNA is contributed from first option sugar, second option phosphate, third option nitrogen base, fourth option all. What is the right answer? What chemical constituent of the nucleic acids contributes the acidic nature? Sugar or phosphate or nitrogen base or all? Yes, answer is a mixed. Somebody said 1, somebody says 2, some are 4. But no one said about the third option. Yes, mixed opinions are there. What is the full form of the P? What is the full form of the P? Actual name of the P is the phosphoric acid. Phosphate molecule found in the form of phosphoric acid. Acid is there. So, acidic nature of nucleic acids in both the DNA and RNA is contributed from P group only, phosphoric acid. There is also one more logic here. Sometimes nucleic acids may exhibit basic nature due to the presence of nitrogen bases. Both acidic and basic together form neutralized form of DNA. Okay, so right answer for this question is second option. Once again, listen to the question. What kind of chemical constituent of a nucleic acid can contribute the acidic nature? Phosphoric acid, because basically it is the acid group. That's why this group causes for acidic nature of nucleic acids, both DNA and RNA. Next, one more question. Name the nitrogen base not present in DNA but present in RNA. Yes, not found in DNA, uracil. Like one more question, name the nitrogen base not found in RNA. Thymine. Remaining bases adenine, guanine, cytosine are common in both the molecules. Okay. Yes, your answering is good. We'll discuss remaining questions in next class. Only one more class is required to complete detailed account of the nucleic acids, detailed account of RNA and DNA structures. Okay. Tomorrow I will explain. Okay, Ram. Bye.
Yes, good morning, students. Hello. Good morning, students. How are you, Charita, Madhumita, Mallikarjun, Deeptika, Ratnapuji, Shravan, Sairaj? How are you? How are you? How are you? Yeah, very good morning to all of you. So yesterday we are done with NEET 2024 and paper was very easy in the biology section. Most of your seniors, your brothers and sisters have scored 360 out of 360. So let us walk in their footsteps and I hope you also get more marks in bio this year. NEET 2026 for you. Done? Guys, is my voice clear, audible? Are you able to follow, listen? Okay, just okay. So today we will be focusing on learning some aspects about, only some, not in depth, basic surface aspects about our skeletal system as well as muscular system also. Okay? Now, there was a simple spelling here, movement. In some textbooks, the word movement was synonymous with motion. So how to define movement or motion? This is movement. Any change in posture, any change in location is considered as movement or motion. So in our body, these movements are mainly because of muscles, sometimes flagella, sometimes cilia, and sometimes also with pseudopodia. All of you know very well that macrophages, which look like amoeba, they move like amoebas. We call that as amoeboid locomotion. And human sperms possess flagellum. So they swim with the help of flagellum. Isn't it? In animal kingdom, like you know about paramecium. Paramecium moves with the help of cilia. Yes, human body also possesses cilia, but in our body, cilia helps in locomotion, but it is not main locomotor structure for us. Okay. But in our body, see guys, this is motion or this is movement. If I just wave my hand like this, my hand is moving, isn't it? My posture is getting moved, my pose is getting changed, isn't it? Then how do you understand the term locomotion? What do you know about this? If I'm waving my forelimb hands, I'm just moving my hands. But this hand movement does not ensure any change in displacement of whole body. But if I move my hind limbs, legs, yes, I'm moving my hind limbs, simultaneously I have also changed entire position. I've also changed my location, isn't it? So movement only. This is movement as well as change in my position, change in my location also. Therefore, we can say that any kind of motion, any kind of movement which can actually change your location, which can actually displace you from one place, to the other place. Change position. All are synonyms. Any movement which can change your location, which can displace you, or which can change your position. These kind of movements are referred as locomotion. Location changing movement. This is movement, not locomotion. This is movement as well as locomotion, isn't it? See, just a movement, but this waving of hand does not ensure whole body displacement, right? But if I move my hind legs, this can easily, these legs are moving as well as they're also changing my location, isn't it? Therefore, one can easily say that all locomotions are movements, isn't it? But all movements are not locomotion, isn't it? All locomotion is because of movement, but all movements does not ensure locomotion. And in our body, flagella helps in movement, cilia helps in movement, amoeba macrophages, they do move, but which kind of structure is allowing us to move as well as simultaneously locomote also? The only structure which allows you to move and locomote simultaneously, those are your muscle cells. Muscle is the structure of your body which is allowing you to perform locomotion. The main tissue responsible for your locomotion was muscle. Muscle was actually derived from mesoderm. Muscle was derived from mesoderm. More than 50 to 60% of your body is composed of mesodermal tissues, mainly due to muscles and connective tissues also. Now, as you already know that muscle is helping in locomotion. Guys, now my question was that, does every muscle of your body, does every muscle tissue, every muscle cell look exactly the same? No. Now, question for you. Tell me, how many types of muscles could be witnessed, could be seen in your body? Your body has how many types of muscle cells or muscle tissues? There are mainly 3 categories. What are those? Skeletal muscles, smooth muscles, and cardiac muscle. Right. Tell me, based on, guys, based on where they were, how they look, and what they do. Based on their location, where they were, how they look, and what they do. Based on the location, structure, and function, we can broadly divide muscle tissues into 3 categories. What are those? Skeletal muscle, then smooth muscle, and then this is cardiac muscle. Okay, skeletal, smooth, and cardiac. Guys, cells of this skeletal muscle cells, they were very elongated, and these cells were arranged parallelly like this. These are very elongated, arranged parallelly, and each cell may have many nucleus. Guys, a question for you. If cell has got more than two nucleus, what do you call them? Multinucleate. So, skeletal muscle cells are elongated, multinucleate. Very good, very good. Now tell me a synonym for, tell me, yes, tell me a synonym for multinucleate spelling. Multinucleate is also synonymously referred as syncytium. Done? Very good. Syncytium. And for you guys, one more synonym I'll give you. Multinucleate or syncytium is also synonymous with a term called as senocytic. Okay? You guys use multinucleate or syncytium or senocytic. It is up to you. Done? And inside this skeletal muscle cells, actin proteins and myosin proteins were arranged in a regular pattern. Guys, actin and myosin proteins are two most important proteins which helps for contraction and relaxation of muscle cells. Okay? Every muscle cell will have them. Every muscle cell was able to perform contraction and relaxation because of these proteins, actin and myosin. In case of skeletal muscle, actin and myosins are arranged in such a way that actin and myosin, not acting. Listen. Guys, students and my children, every muscle cell will possess actin, myosin, and these are helpful for contraction and relaxation. Every muscle cell will have this. In case of skeletal muscle, actin and myosin are arranged in such a way that we can see a pattern like this, like a zebra crossing. Regularly, disciplinedly assembled inside muscle cells like this. What are these? Actins, myosins. Like this. See? Because of arrangement of actin and myosin like that, if you observe this muscle cell under microscope, you will see lines. These lines are referred as stripes, or we can also call them as striations. Guys, you can call them stripes or striations, up to you. Therefore, skeletal muscle is also striated in appearance. How they look, I told you, right? And yes, first we learn about skeletal, then we can jump on to smooth and cardiac. I'll tell reference also, don't worry. Guys, skeletal muscle is elongated. Cells are elongated, multinucleated and possess many striations. Okay. Coming to smooth muscles, these are spindle shaped like this. Bundles. These are spindle shaped with only single nucleus, that too at center. So please be careful with this. Smooth muscles have spindle shaped, and spindle shaped is synonymous with fusiform. You can call them spindle shaped as well as fusiform. In case of smooth muscles, yes, smooth muscle also has actin myosin. Smooth muscles also exhibit contraction, relaxation, but inside smooth muscles, there is no regular pattern wise arrangement of actin myosin. No. Actin and myosin are very randomly arranged. Therefore, you do not get to see any striations in them. As actin and myosin are unevenly, randomly arranged in them, smooth muscles does not show you any kind of striations. Therefore, these are non-striated, non-striped. Is it clear this point, till this point, guys? No striations in smooth muscles because actin and myosin are randomly, unevenly arranged. Done. Coming to cardiac muscles. So, in which location of your body do you say cardiac muscle? Tell me. The exact location, the only organ of your body, where you say cardiac muscle. Easy question. Try answering that. The only organ of your body, where do you see cardiac muscle tissue? Heart. Very good. Then yes, correct. Only heart will exhibit cardio, heart. But under microscope, how does this tissue looks like? Under microscope, cardiac muscle tissue may look like cylindrical, but they have got branches also. See, there are many branches. Like this. Yes, we'll get into functional details slowly. First, you need to understand structural relations. Yeah, we'll go in deep. Not now. First, please learn basics, then we can move on to advanced technology. Done? Sairaj, yes, no fatigue, not now. Wait for some time. Yes. Okay, see? Guys, cardiac muscles, they look like very short cylinders, short, cylindrical, and almost all of them are exhibiting branches, therefore branched. Short, cylindrical, and branched. Done. And like skeletal muscle, even in cardiac muscle also, actin and myosin are arranged in a pattern. Therefore, even inside cardiac muscle, we can see striations like this. Cardiac muscle also exhibits striations. Therefore, we can say that cardiac muscle is also striped muscle. Yes, cardiac is striped. Very good, correct. It is striated. Even cardiac muscle also exhibited striations. Correct. But in cardiac muscle, minimum 1 nucleus to maximum 2 nucleus per cell. It is either single nucleated or binucleated. Possible. See guys, smooth or single nucleated. Cardiac muscles may have 1 or 2 nuclei, but striped muscle may have many nuclei. Multinucleate, uninucleate, uni or binucleated. Okay, uni or binucleated. Done. Now, these are structural aspects of muscle cell. Coming to their functional role. See guys, skeletal muscle, smooth muscle and cardiac. Skeletal muscle is always attached to bone or cartilage. But smooth muscles are never attached to bone or cartilage. No attachments to bones or cartilage. Even cardiac is also never attached to bones or cartilage. No. B stands for bone, C stands for cartilage. Only skeletal muscle was found attached to bone or cartilage. Neither smooth muscle nor cardiac could be found attached to bone or cartilage. No. Therefore, this can be controlled by you easily. This is willful control. You can control this. These muscles listen to your brain. As you say, they will act. You can voluntarily control them. Voluntary. Reason why? Because these are attached to bones. But smooth muscle not attached to bone, you cannot control them voluntarily. They were involuntarily controlled. Your involvement is zero for this. And we can say that autonomous, automatically controlled. Same time, even cardiac is also not attached to bone, therefore it was also involuntary. Synonym for involuntary, automatic, autonomous. See, striated, voluntary, non-striated, involuntary, striated but involuntary, isn't it? This is striated, this is non-striated, and this is also striated. Done? Now we can frame 3 questions here. Accepted? Which muscle is striated and voluntary? Skeletal muscle. Which muscle is striated and voluntary? Skeletal muscle. Which muscle is striated but involuntary? What is that? Which is striated but involuntary nature? Cardiac. No striations. Very good, very good. Non-striated, involuntary. Smooth muscle. Okay. Guys, we call them skeletal muscle. We call this as skeletal muscle cell or voluntary muscle, up to you. Whereas smooth can be told as smooth muscle. If we say involuntary here, that will lead to some confusion. Therefore, as these muscles are found inside your body tissue, smooth body tissues, we can say this smooth muscle also as visceral muscles. So skeletal is voluntary muscle, smooth muscle is also called as visceral muscle. Cardiac. No other names for cardiac, that's it. Heart muscles. Heart muscles, okay? Till now we learnt about structural details and functional details only. Now you will give me some examples of these locations. Now tell me one example of skeletal voluntary muscle you know. Give me one example, guys. Skeletal voluntary muscle which is controlled by you. You can willfully control it. One example of skeletal muscle. Try this. Give me one example of skeletal muscle. Yeah, leg muscles, hand muscles, limbs, correct. Attached to bone. Yes, Garuda, yes, it is attached to bone. One more. Biceps, very good. Triceps, very good. Biceps and triceps both were voluntary, yes. One more. Your eyelids, correct? Eyelids, isn't it? And tongue. How could you forget this? Tongue, isn't it? Tongue is voluntary. Yeah, quadriceps, correct. Yes, thigh muscles. Yes. Very good, Sairaj. Accepted. All these are examples of voluntary muscles. Lips, correct. Lips are voluntary. Fingers, yes, voluntary. Very good. Accepted. Very good. Now give me only one example of cardiac muscle. No need to ask anyone. That was obviously the only place you can find cardiac muscle was. Yeah, neck is also striated, correct. Heart. Now, question. So, cardiac muscles consist of striations, yes or no? Cardiac muscles consist of striations, yes or no? Yes. Cardiac muscles are voluntary, yes or no? Cardiac muscles are voluntary, yes or no? No. They're actually involuntary, isn't it? Cardiac muscles are also involuntary. Now, this is very interesting aspect of our class. Smooth muscle. Guys, you swallow food. Once food enters esophagus, till food reaches rectum after digestion, esophagus, small intestine, large intestine, In large intestine, you can see cecum, rectum, cecum, colon, rectum. Starting from your esophagus till the food gets digested, and undigested food will be sent to the rectum. Entire food will be pushed downwards by smooth muscles, isn't it? Simultaneously, increase or decrease in a blood pressure is because of constriction and dilation of blood vessels. Dilation means like this, relaxation. Constriction is like this, contraction. Blood pressure is actually controlled by constriction and dilation of blood vessel. Who does that again? Smooth muscle, isn't it? So entire alimentary canal and entire blood vascular system and blood vessels except heart, all blood vessels will consist of smooth muscle. And even your pupil, eye has a circular aspect thing and has a cornea. You know this, behind cornea you can see pupil. If you go in dark, pupil dilates. If you come near light, pupil constricts. So pupillary dilation, pupillary constriction is also done by smooth muscles. Guys, entire alimentary canal, blood arteries and veins, pupillary dilation, constriction, all were done by smooth muscles. Done. Now there was a practical question for you. If you come here, I will give you a microscope and muscle tissue, possibly chicken muscle tissue. Could you easily observe muscle tissue under microscope and decide whether it is voluntary or smooth or cardiac? Is it possible? Is it possible to decide whether it is a voluntary, smooth or cardiac just by looking it under microscope? Yes or no? Yes, Sarah. Yes, Tikshana. Very good. Yes, confirmly yes. Because if you see muscle cells, if they are long cylinders, no branches, striations are there, voluntary. If the short cylinders, branched striations, cardiac. No striations, no branches, spindle shaped, smooth muscles, isn't it? The main motto behind learning their structural differences is to discover them under microscope. That is how they look under microscope, isn't it? The first criteria for classification was that how they look, morphological classification. Then we did for anatomical and functional classification. This is morphological. How do they look? This is how they look. So unbranched, no branches, long with striations, skeletal. Short cylinders, branches are there and striations are also there, cardiac. No striations, no branches, spindle shape with single nucleus, smooth. Done. Now, among skeletal, smooth and cardiac, the main tissue responsible for locomotion was one and only skeletal muscle. The only muscle which helps you to perform locomotion. Guys, every tissue can help you for movement, but I'm speaking about locomotion only. The only tissue which helps in locomotion was skeletal muscle only. Only skeletal muscle can help you for locomotion. Good. Now, so it was very clear and obvious that we need to learn in detail about skeletal muscle. Let us zoom inside cytoplasm. let us look at actin myosin, and let us also learn how this actin and myosin are helping these muscles to contract as well as relax. Okay? Guys, am I clear till this point? Understood structural and functional aspects of muscle cells. Done, Vaishnavi? Balu? Sushant? Rishita, locomotion means I do some movement. See, this is a movement. I am moving my hands, but my body is not changing its location. I didn't move anywhere. I'm here moving. Then what do we have locomotion? I move my legs now. I change my location also. Isn't it? Therefore, every movement doesn't help you to locomote, but every locomotion is because of movement. So all locomotions are movements, but all movements are not locomotions. Understood, Rishita? So, done? Change in posture is movement. Change in position is locomotion. Done? Done? So the main tissue responsible for locomotion is skeletal. Question for you now. Question 1. Skeletal muscle is striated or non-striated? Answer fast, rapid. Skeletal is striated or non-striated? Yes, striated. Very good, Balu. Good. Next question. Skeletal muscle is branched or unbranched? Very correct. Unbranched. Do they have single nucleus or many nucleus? Do they have single nuclei or many nuclei? Yes, many, multinucleated. Are these skeletal muscles voluntary or involuntary? Which means, are they attached to bone or not? Yes, they are voluntary in nature. See, correct. So if we zoom a muscle tissue, see, here is a bone. Very famous picture we see in Tom and Jerry or any cartoon shows. A dog will be there which is always chasing for this bone, right? A very simple bone. And this bone has a tissue attached to it like this. And to this tissue, you can see attachment of a muscle. Now question was that, what is this organ? This is a bone. What was this organ? It's a muscle. Question now. Which tissue is acting like a bridge between bone and muscle? That was tendon. As I told you, skeletal muscle is always attached to bone. Therefore, energy of muscle can be given to bone, and bones can exhibit movement which can help in locomotion. Done? So bone, tendon, muscle. Guys, entire muscle, I'm showing you a simple picture, is muscle tissue. If you zoom inside muscle tissue, they consist of many muscular bundles like this. Muscle tissue consists of muscle bundles. These muscular bundles are called as, muscle bundles can be known as fascicles. There were many fascicles, right? Muscle encloses many muscle bundles. Muscle bundles are called as muscle fascicles. And all these muscle fascicles were actually joined together with the help of connective tissue also. There is the tissue enclosing these fascicles like this. Okay, what is this connective tissue which is enclosing fascicles? It is called as fascia. Tissue enclosing fascicles was fascia. Now, if you zoom furthermore, inside each fascicle, you can see many muscle fibers. Inside each fascicle, sir, there are many muscle fibers. This is a muscle fiber. Muscle fiber is synonymous with muscle cell. See? Picture. Muscle tissue has fascicles. Fascicles have muscle fibers. Muscle fibers are synonymous with muscle cells. Okay? Is it okay till this point, guys? Just learn basic thing. Do not get into depth of this concept. Just stick to these three parts: muscle, fascicles, muscle cells. Outermost muscle, level 1 muscle, level 2 fascicle, level 3 muscle cell. Done? Okay. So tomorrow we'll continue from this point and we'll learn about how do this actin and myosin actually help in contraction and relaxation. Okay? Shall we meet tomorrow? So go and have lunch. Stay indoors, stay hydrated. Done? Thank you.